也欢迎你回来到我这来看一看。你常常回来，
我们不，我们不能去看你啊！
你可以来看我了，又不是不能看，
私人
可以
私人
看我。
私人去看，私人去看。
对啊，你来看我嘛。对，
对，
欢迎你们来。
是。
慧荣老师来了没有啊？慧荣老师，
他好像
我
那个会议号登录不进来，在那跟我联
哦
系，然后对
哦
我说让他试一下，因为 Room 有时候需要你去更新一下 update，然后才让进。所以我今
我不认，
天我说我也不 sure，说要是我 Room 掉线的话就靠你了，助理。
网络挺好，没关系的，没问题。
嗯。对，我们再多等2分钟，好像对尹老师好像还没上来。
丽涵上来了，不好意思，我以为是搞成那个那个叫啥，我们的另外一个 app，对不对？我已经上了，谢谢啊。
那我们差不多可以开始了，就是
是，
9点。对，距离你把尹老师变成 co-host 哦，已经
我设置
可以了。
成了，
嗯嗯。
那我们开始哈。对。Good morning to friends in China and good evening to friends in United States. Welcome to the 231st online forum of Metabolic H Talk. My name is Li Hengwang. I'm an investigator from Peking University Health Science Center. Together with Dr. Julie Bai from UT Houston Health Science Center, we will host today's event. Today, we are honored to have Professor Yang Xia with us. Dr. Yang Xia is a National Distinguished Professor at Xiangya Hospital of Central South University. She is a leading expert in metabolomics and hypoxia research. She has developed innovative high-throughput metabolomics and isotopic tracing methods to decode metabolic dynamics, uncovering the uh, erythrocyte hypoxia sensor mechanism that links energy metabolism to ischemic disease. Her work identifies critical biomarkers and therapeutic targets across life stages, from pregnancy to aging. With over $21 million in funding, she has published more than 130 papers in the top journals such as Nature Medicine, Cell Circulation, JCI. She also won a lot of awards, including U.S. Lyndon Baines Johnson Presidential Award, the American Heart Association Innovation Award, and the NIH Director's Challenge Prize. Additionally, she holds leadership roles in international scientific societies, advancing research in metabolic health and disease. In addition, we also invited a few panelists to join today's seminar. Um, including Dr. Su Nen Fu. He is a principal investigator from Guangzhou laboratory who is an expert on studying metabolic fluxes and proposed the metabolic roundabout hypothesis. Dr. Ai Juan Qu from Capital University. She is working on the mechanisms of metabolic remodeling associated with cardiovascular disease. And Dr. Hui Yongyin, a professor from City University of Hong Kong, he focuses on the redox regulation of glucose and lipid metabolism. And Dr. Shu Hai Ling, who is a professor from Xiamen University, he is an expert on the metabolomics and also the mechanism study of many human disease, including liver, lung cancer, and COVID, and so on. So welcome. The title of today's talk is erythrocyte hypoxia sensor, a key metabolic regulator of disease, aging, and longevity. So Dr. Xia, now the stage is yours.
Thank you, Li Heng, for such a wonderful introduction. And also, I'm very happy to have opportunity to present almost 10 years of my work in less than 1 hour. And also appreciate all the panelists to take a busy time to come weekend to listen such my seminar talk. So the topic, as Li Heng mentioned, is erythrocyte hypoxia sensor, a key metabolic regulator of disease, aging, and longevity. So I would like to first talk a little bit about hypoxia. So you may have heard the Nobel Prize honor. The owner is about HIF response induced factors related to hypoxia. And hypoxia indeed is the dangerous condition involved in tissue damage. When we face hypoxia, there are two types of major hypoxia. One is systemic hypoxia. For example, physiologically, you go to high altitude or go to deep sea ocean diving, you are facing the low concentration of hypoxia. Your systemic whole body faces the hypoxia. And also has a pathological hypoxia, such as sickle cell anemia or thalassemia. So you have a mutation in hemoglobin, then you face the red blood cell dysfunction, then all of the organ faces the tissue hypoxia. We also have pathological tissue hypoxia. It can involve almost every organ, such as stroke, you have brain damage, and heart failure, cardiac attack, and hemorrhage, stroke, etc., and including other like preeclampsia, pregnancy-related hypoxia, aging, and cancer, etc. So hypoxia indeed is a very dangerous condition. However, red blood cell, as the only cell deliver oxygen, but its function and the regulation in hypoxia is poorly studied. So what's the feature of a red blood cell? So red blood cells are the key hypoxia sensor in our body. It is the most abundant and a very dynamic cell type. It almost like occupy 80% of a host cells. So 20% cells are other cell type, including brain and the neuron and other cell. 80% of our circulating and the total cell are red blood cell. So also red blood very dynamic. Every second we produce 2.4 million new red blood cell. It requires a lot of energy and nutrient to make a new red blood cell to support the red blood cell function. And also red blood cell very simple, no nuclei, no intracellular organelle, except the proteasome. And it no new protein synthesis, no HIF-dependent transcription regulation. It mainly depends on post-translational modification to control the protein stability and the metabolism. And it also very important to know red blood cell not only deliver oxygen, but also very sensitive to hypoxia. When it is sensitive, hypoxia can release more oxygen. And it also has a membrane for hypoxia. So based on all these features, and my laboratory has worked for almost 10 years to address two key questions: how red blood cells sense the hypoxia and enhance its oxygen delivery capacity to counter tissue hypoxia, how red blood cell maintains its memory to hypoxia. So to address this question, my laboratory has been very fortunate, involved 6 countries, 25 scientists collaborative work involved to answer the major question, use multi-omic study to understand the physiological response of human to high altitude. To address this question, the human subject design is like this. We recruit 21 young male and female, half female, half male college student, 21-year-old. And the experiment design is like bringing these 21 young college students from sea level to the high altitude. And then we let them to stay high altitude up to 16 days. And we connect a different time point from sea level to day 1, day 2, and day 7, different time point of our red blood cell, and also monitor their physiological and red blood cell function. And in order to answer whether they maintain the high altitude memory, and we also bring them down one day. After 16 days, we bring them to sea level again, and let them stay 7 to 21 days at the sea level, then bring them back to high altitude for the second time. We connect the second time, day 1, red blood cell function and other physiological parameters. And after we design this experiment, my laboratory work with Angelo D. Alexander, is my good friend in UC Colorado, University of Colorado. We did a metabolomic study. We do find high altitude induced red blood cell, a lot of metabolic change. And the 3 major metabolic changes: purine metabolism, sphingolipid, and glucose metabolism. And later on, we accurately measured the 2 metabolite, 2,3-BPG, and sphingosine-1 phosphate, and its related enzyme, SPHK1 activity to produce S1P. We do find the time-dependent increase in response to high altitude. So I would like to take this opportunity to briefly introduce what's the 2,3-BPG. Basically, 2,3-BPG very unique metabolite, largely produced in red blood cell by glycolysis. So it's through one of a very unique shunt named the rumponuclein shunt. Usually glycolysis will go all the way from 1,3-BPG to 3-PG to ATP to lactate production. But in red blood cell contain very important enzyme named the 2,3-BPG mutase can convert 1,3-BPG to 2,3-BPG, and this is a very important metabolite. This metabolite, the concentration is around 5 millimolar concentration, even closer to hemoglobin concentration in red blood cell. Why our body produces such a high concentration of 2,3-BPG? And it is known that the 2,3-BPG as a small molecule compounding with the central water cavity of tetramer of hemoglobin. And this bonding can cause conformation changing of hemoglobin, and this trigger the oxygen release. Therefore, it is known in the textbook, 2,3-BPG is the most abundant metabolite in red blood cell compounded to central water cavity of hemoglobin, trigger the oxygen release. And additionally, S1P is also a very unique metabolite. It is known the S1P can produce in red blood cell by sphingosine kinase. And also S1P cannot further degraded by the red blood cell because due to lack of its degrading enzyme. Therefore, when S1P produced in red blood cell is largely will store there or released through one of the transporter and to the circulation. So red blood cell is considered as the most prominent cell type to produce S1P extracellularly provide plasma S1P. S1P is a single molecule, can signaling through the 5 S1P receptor function regarding the immune response and the cellular function, fibrosis, etc., even in the cancer. However, S1P in red blood cell function remain unclear. So to study what's the function of S1P in red blood cell, particularly in response to high altitude. We generate ERISA-specific knockout SPHK1, and we mimic human high altitude to expose the control and mice to the high altitude hypoxia. Then we found indeed in Y type the SPHK1 activity, S1P and 2,3-BPG and P50 all increased in mice similar like a human climb to a high altitude. However, knockout SPHK1, we do not see the SPHK1, S1P, 2,3-BPG, and P50 induction and as Y-type mice. So this result indicate SPHK1 is essential for hypoxia-mediated elevation of ERISA S1P and 2,3-BPG and P50 oxygen release capacity. Later on, we also validated the tissue level without ERISA SPHK1 as the tissue such as kidney and has showed heart, showed more hypoxia. This hypoxia probe indicated the SPHK1 mediate oxygen release likely through 2,3-BPG is very important to counter tissue hypoxia. So then what's the potential mechanism and how the SPHK1 cause 2,3-BPG induction is the next question we would like to address. So in order to address this question, I would like to share with a little bit of literature about a unique feature of red blood cell metabolism. So as mentioned, the glucose metabolized generate large amount of 2,3-BPG to regulate oxygen delivery. But the red blood cell carry oxygen, never use oxygen. So oxygen, it also can potentially dangerous for oxidative stress. To find regulate oxygen delivery and anti-oxidant stress mediated by oxygen. And the red blood cell very smart, use a lot of glucose to generate go through the pentophosphate pathway to generate NADPH to couple the glutathione to anti-oxidant stress. Therefore, red blood cell has finely regulated glucose metabolism and to maintain its oxygen delivery and also anti-oxidant stress. So what's the potential known mechanism to regulate such a fine regulation? It's very unique and sophisticated. In red blood cell, as I mentioned, it cannot synthesize new protein, and the modification of the enzyme is not only by post-translational modification, it also by the membrane trafficking. So for example, in the low MOX condition, glucose metabolism is largely regulated by the enzyme trafficking. So in low MOX condition, one of the key enzymes usually you use as a loading control, GABA-DH actually in red blood cell cannot perfectly as a loading control. Usually it's localized on the membrane. And when the low MOXia, we don't release that much oxygen. So because red blood cell carry 4 oxygen tetramer, only release 1 or 2. So at that time, a lot of oxygen stored in red blood cell at the low MOX condition. So GABA-DH at this time is anchored at membrane. So the glucose metabolic is regulated by largely go to pentophosphate instead of glycolysis due to this GABA-DH anchored at membrane in the band 3. So this sophisticated membrane trafficking or anchored on the membrane of GABA-DH preferentially mediate glucose metabolism shunted pentophosphate instead of glycolysis. This actually is important for the red blood cell anti-oxidant stress because they don't relieve enough oxygen and they stored some oxygen at the low MOX condition. However, in response to hypoxia, red blood cell quickly deoxyhemoglobin become more deoxyhemoglobin, and the deoxyhemoglobin will traffic to the membrane and to anchor to the band 3. At this time, we trigger the GABA-DH release from membrane to the cytosol, and this release GABA-DH will trigger the glucose metabolism shunted glycolysis instead of pentophosphate. This will trigger the more likely glycolysis at the same time produce more 2,3-BPG and trigger the oxygen release to counteract tissue hypoxia. Therefore, red blood cell has a very sophisticated membrane trafficking of the GABA DH and also deoxyhemoglobin and can trafficking to the membrane, this switching and can make the metabolic of glucose reprogramming and trigger the glycolysis to respond to hypoxia. Based on our discovery, we found ACE1P actually can increase the 2,3-BPG, increase the oxygen release as shown previous slides, and it lead us to further hypothesize that maybe ACE1P is one of the unknown or missing component in red blood cell produce such a high amount, particularly in response to hypoxia, can interact with deoxyhemoglobin and bring it to the membrane and therefore trigger the GABA DH release and furthermore trigger the glycolysis, trigger the 2,3-P production, trigger the P50. To test this hypothesis, we first test whether when we expose wild-type ACPH-K1 mice to the hypoxia, we can see the GABA DH activity and the change in these mutant mice. And we found in wild-type mice, the GABA DH is nicely responsive to hypoxia. Activity is induced. However, lockout the ACPH-K1, such activity is blunted. And at the same time, we use Western and in immunostaining, we found the GABA DH. When we lock out ACPH-K1, the release or increase the GABA DH release from membrane to the cytosol is substantially reduced. Basically, when we wild type, you can see the GABA DH normally in the low oxygen is entered the membrane. But when we in response hypoxia, GABA DH release to cytosol, trigger the glycolysis. But when we lock the ACPH-K1, GABA DH failed to release cytosol, still enter the membrane. So these data indicate that ACE1P is very important to response to GABA DH release and trigger the glycolysis in response to hypoxia in red blood cell. And later on, we collaborate with my collaborator, Martin Saffel, and use the co-crystal structure and demonstrate the ACE1P indeed can binding with deoxyhemoglobin co-crystal with 2,3-BPG together at 1.9 angstrom. So this study and demonstrated by the function, genetic, and crystal structure level indicate that ACE1P is a very important single molecule, can interact with deoxyhemoglobin and trigger the glycolytic enzyme release from membrane to the cytosol, trigger the glycolysis, trigger the 2,3-BPG production, trigger the oxygen release, counteract tissue hypoxia. Later on, we also reviewed the non-signaling pathway, adenosine also important through AMPK and the BPG mutase phosphorylation and regulate glucose metabolism, trigger the 2,3-BPG oxygen delivery and lower tissue hypoxia in response to hypoxia. So this study and using the high altitude, the human work, genetic and crystal structure and the cellular level, we bring the new concept. Red blood cell have HIF independent, but the glucose metabolic regulator hypoxia sensor. And the red blood cell hypoxia sensor is a new target for combating hypoxia tissue damage. And at the same time, I mentioned that we're also very fascinated by hypoxia red blood cell membrane. We do find that when we bring the human to the first time to high altitude day 1, almost everybody has acute mountain sickness. You can see from here the score. But when they climb the second time and on day 1, almost similar like day 16, and they quickly adapted no acute mountain sickness. And down the road, we found the plasma adenosine and also soluble CD73 to produce the adenosine, the enzyme is significant, quickly, rapidly elevated at the second time, day 1, compared to the first time high altitude, day 1. So follow this human work, we use the genetic and the cellular human, also pharmacological study, and further demonstrate this quickly rapid increase of adenosine through the soluble CD7 activity is partially, potentially important purinergic hypoxia response and is the key for rapid adaptation for the second ascending to the high altitude. And after we study the normal physiological hypoxia, I would like to share with you one of the disease my laboratory has studied for almost 10 years is preeclampsia. And preeclampsia is a disease of related our new life beginning and also related pregnancy. It's a disease of pregnancy. It affects 3 to 5% of first pregnancy, and also in the high altitude, 20 to 30% preeclampsia occurred, and much higher than the normal sea level. So also it's indicated it's likely related hypoxia. And also clinical definition is appear after the 20th week of gestation. Symptoms include hypertension and proteinuria is kidney damage too. So the treatment is very poor in the clinic, except like resting and other related aspirin and such kind of a treatment, but it's really no good effective treatment. The only treatment we so far is deliver fetus and placenta. Once deliver, the preeclampsia is gradually gone. The cause is unclear. So my laboratory has used the screening system, use cellular level screening system, and has identified in the preeclampsia patient, that's an allolistic antibody, is IgG type. It can specifically function through the AT1 receptor, and it can function as agonist at a multiple cellular level, such as cardiomyocyte, can increase the contraction rate through AT1 receptor activation, and can increase human trophil cell line, can increase the PI1, NADPH oxidase, soluble fluid secretion, and then also can increase vascular smooth muscle tissue factor and human messenger cell PI1 secretion. All these factors are related pathogenic feature of preeclampsia. So this in vitro screening system point out in the preeclampsia patient, there's a circulating agonistic or activating autoantibody can activate the AT1 receptor and likely contribute to pathophysiology of preeclampsia. And down the road, we also use competition assay as screening this AT1 agonistic antibody, AT1A, functionally likely binding to the second actual loop of the AT1 receptor. Because when we use 7 amino acid peptide, we can specifically block AT1 mediated cellular activation and induce downstream pathogenic factor as shown in the early slides. So these data indicate AT1A specifically recognizes 7 amino acid epitope on the second actual loop of AT1 receptor. Now, next, we would like to address whether this autoantibody indeed in vivoli can induce the preeclampsia feature. To address this question, we purify the AT1A from the preeclampsia patient, inject the pregnant mice. We do find that the injection of AT1A induces 2 major features of preeclampsia, hypertension and proteinuria. More importantly, these 2 features can blocked by losartan, which is AT1 specific receptor antagonist. It indicates this autoantibody likely function through AT1 receptor activation. At the same time, it also can neutralize or blocked by 7 amino acid epitope of the peptide. It indicates AT1A functions through actual interact with the actual cellular loop of the AT1 receptor, that 7 amino acid peptide. So from here, we demonstrate autoantibody from the preeclampsia patient can induce preeclampsia feature, which is a potential pathogenic immune factor contributor to preeclampsia. It also raises a new concept. Preeclampsia is a gestation-induced autoimmune condition. So this work has been validated by many labs nationally and internationally. Down the road, we're also wondering whether this detrimental autoantibody can cross from maternal circulation to the fetal circulation, as we know the antibody can cross. So we first measured the cord blood. We indeed found the autoantibody AT1 can cross maternal circulation to the fetal circulation and exists in the cord blood. And we also use our double transfer experimental model, and we found the AT1 injection not only induce abnormal placenta, but also induce small fetus. So this work has been published in the JEM, demonstrate this autoantibody is detrimental not only to the maternal generator P feature, but also detrimental to induce the fetal growth restriction. So next bigger question, and for my laboratory has addressed and also has continued to address now, how is the AT1 generated in preeclampsia? So my team and multiple other teams, including the Mississippi and Germany and multiple group, has devoted time to demonstrate placenta hypoxia, such as restricted uterine placenta perfusion, and also infuse the multiple cytokines such as TNF-alpha, light, interleukin 17, and the cause maternal immune response, all can directly induce autoimmune and induce AT1A production and induce PE feature through the placenta hypoxia, metabolic impairment, cellular injury, and down through the maternal immune response, cause the allographic autoantigen generation, cause autoimmune, and cause AT1A production and PE feature. But how the placenta hypoxia is initiated remain unclear. So my laboratory has wondering whether the red blood cell function or metabolic has been changed in the preeclampsia, cause the placenta hypoxia. To address this question, I was very lucky and try to continue study the purinergic hypoxia response. So as I mentioned in the early work, we talked about the adenosine is very sensitive to hypoxia. It has very sophisticated metabolic purinergic regulation. So you normally ATP store in red blood in every single cell as energy, but in response to hypoxia stress, ATP can release extracellularly through the two key enzymes, CD39, CD73, generate ATP to AMP and further generate adenosine by dephosphorylation. So ATP, the 3-phosphate gone, generate adenosine. Adenosine can further convert it to inosine too. And also adenosine can go through the equilibrium transporter and to be eliminated and then go inside of the cell and by ENT transporter. At the same time, the well-known function of adenosine is 3,4-adenosine receptor as a signaling cascade to regulate vasodilation, anti-inflammation, anti-vascular leakage, and even red blood cell oxygen delivery. However, erythrocyte ENT, whether play important function, remain unclear. So my laboratory generated erythrocyte-specific ENT transporter deficient mice, and one of a very talented postdoc in my laboratory from Tokyo University has used these mice, unexpectedly found one the only maternal deletion of the red blood cell ENT, and this fetus much smaller than normal mice. And we also further found this placenta also substantially reduced. So then later on, we used the metabolomic and the enzyme and the function to demonstrate indeed purinergic signaling cascade has been substantially reduced by lockout erythrocyte ENT, and this led us to further demonstrate MPK activity also substantially reduced in this specific lockout mice, and also the BPG mutase substantially reduced, 2,3-BPG and the P50 reduced, and also the antioxidant stress like a GSH or GSG ratio reduced, result increased the ROS. So this data demonstrate erythrocyte-specific ENT play very important role to regulate MPK, BPG mutase-dependent oxygen delivery and anti-ROS. How about the placenta feature? So we do find that when we look at the ERISA ENT, the placenta more hypoxia. Metabolically, very strikingly, we found that when we look at the ERISA ENT, the amino acid in the placenta is substantially reduced, almost every amino acid. However, the plasma amino acid is substantially accumulated. So this implicates in the ENT1 lockout in the red blood cell controlled amino acid transport from plasma to the fetus through the placenta. Indeed, we found that the placenta, one of the transporter, which is responsible for, this is basically transport for the amino acid LAT1 is a very important transport, substantially reduced. And later on, we used the flux experiment labeled amino acid. We found that the amino acid in the placenta in the lockout is substantially reduced and accumulate in the plasma. So these demonstrate the ERISA ENT deficiency promotes placenta hypoxia, reduce amino acid transporter, and is uptake from the plasma to the fetal circulation. So this work has been published and demonstrated the red blood cell as a very important regulator of oxygen, and through this sophisticated ENT transporter, and play very important function, regulator cascade and oxygen release, and then down the road lead downward or maintain the amino acid transporter to maintain the fetal circulation. If red blood cell function disrupted, then this will lead the hypoxia, then lead amino acid transporter reduce, then therefore the plasma to the fetal, maternal plasma to the fetal circulation of the amino acid is substantially reduced, lead to the fetal growth restriction. So here we demonstrate red blood cells major response of oxygen regulate the placenta hypoxia in the preeclampsia or likely in the fetal growth restriction. And this is a major cause the placenta hypoxia, cause the metabolic impairment, cause autoimmunity, cause the preeclampsia development. After I talked the preeclampsia, I want to share with some most recent work related to aging. This is the last part I will share with you. So this is what the work, as everybody realize, aging is a global problem and a challenge. And aging affects every organ. Substantial work has been devoted the effort to study the tissue hypoxia at the tissue level, every single organ and the metabolic disorder. But it has been now facilitated by animal work, even some human work. Combine the young mice with old mice, and they found that the old mice were rejuvenate, 返老还童. Then there's always speculated that there's a mysterious protein or small molecule for rejuvenation, for the people live longer or anti-aging. So most of the study focus on the circulating metabolite or small molecule. However, as the most abundant and also responsible oxygen release, whether such a red blood cell play important function during the aging or during the longevity remain very poorly studied, is extremely overlooked. So to answer this question, and I was very fortunately and has opportunity to isolate almost 800 and aging and longevity cohort human study in Hunan province. So this including young, middle-aged, older group and the longevity group from the 21 to 100-year-old, 1,800 different human subject study. And to address the question whether red blood cell play function, we first took a lot of effort to measure all these 800, each individual, half male and half female, and their red blood cell oxygen release. We do find, as you can appreciate it, compared to the young, during the aging, the red blood cell function is gradually declined, actually started as early as around 50 or 30-year-old, that gradually declined until 89. But when the longevity group, their red blood cell function release oxygen not only not decline as the aging, but maintain the close like young people. So this indicates during the normal aging, our red blood cells gradually decline, but the longevity individual has more young like a feature oxygen release capacity. At the same time, we compared their inflammation and the liver function and the metabolic as the glucose and the LDL and the HDL. We found that during the aging, this feature gradually changed, but this longevity group has a useful oxygen delivery capacity, but also has immune liver function and metabolic function like young people. So this demonstrated longevity people live longer, potentially has higher young like oxygen deliver and the young immune liver function and the metabolic function. To understand the metabolic feature behind, we conducted both plasma and the red blood cell 16,000 and the plasma 16,000 sample metabolic screening. We found the longevity individual has the most significant change in purine, glucose, and amino acid metabolism. And later on, we divide all these metabolic feature to young aging feature or rejuvenation signature based on their trajectory. So we do find one of the two of the metabolite I have mentioned is 2,3-BPG and the inosine that basically decrease during the aging, but has a higher level in the longevity red blood cell, basically reduce the during the aging, but in the longevity individual, their level much higher than the aging people close to the young people. So this implicates this likely can be potential rejuvenating factor responsible for the higher or use like oxygen deliver capacity in longevity individual. To test this hypothesis, recently we performed both human and mouse in vitro red blood cell experiment. So we basically isolating red blood cell and the mouse red blood cell, treated the inosine with this red blood cell. We found that indeed the inosine can increase the oxygen release in a dose-dependent manner, and also can anti-ROS induction, anti-ROS in a dose-dependent manner. So furthermore, we want to know whether inosine function through the receptor or through the transporter uptake by ENT transporter. We use the inhibitor-specific ENT and A2B receptor. We found the inosine basically function through ENT transporter because this induced P50 can blocked by ENT specific transporter, but not by A2B transporter. This work has been validated by mouse red blood cell culture. So from here, we demonstrated that inosine increase or higher in the longevity red blood cell likely function and through the ENT transporter, but not through A2B specific receptor. So to further validate this in vitro work, we use this ERISA specific lockout to compare 2 months old, 6 months old red blood cell function and also their cognitive function. As you can see, at a young age, the mice, the P50 and the cognitive function is similar between the normal and the mutant mice. But when they become older and the oxygen release capacity and the cognitive function and start to decline and much accelerated in the ERISA specific lockout. So these mutant mice demonstrate ERISA ENT lockout mice reduce oxygen release and accelerate cognitive decline in aging mice, indicate the ERISA ENT mediated inosine uptake is extremely important. It's one of the anti-aging important transporter and to uptake inosine. Later on, we're wondering whether we can rejuvenate aged mice by supply the inosine. So we found the one we inject the inosine to the 16 months old mice. We found that we not only can enhance the oxygen release, but we also can increase muscle strength in 16-month-old mice after 1 month treatment. And we also can increase the cognitive function in aging mice after 1 month innocent treatment. At the same time, we go to look at the hypoxia level. We find the inosine can decrease the hypoxia level in the hippocampus in aging mice. So lastly, we wondering how the inosine work. So we compare the young and aged mice. We use the label the inosine and conduct the in vitro red blood cell flux experiment. We found the inosine acts basically in aging the human or aging the mice. Inosine is reduced in the red blood cell, is largely and may potentially contribute to the P50 reduction and during the aging. However, in the longevity, when they have a high level of inosine, inosine can transport uptake by ENT transporter and then can metabolize to the ribose 1 phosphate. And this can function as additional carbon source to provide the fuel to shunt the PPP pathway, glycolysis, and this can increase the 2,3-BP, ATP, NADPH, and then to trigger the oxygen release, to anti-ROS, to contact tissue hypoxia, brain damage, and the cognitive decline during the aging, and make the mice and the people live better and a longer health span. So I want to emphasize there's a blue 5 balloon zone in whole world, and this 5 balloon zone is named the world longest lived population. They're in the United States, in the Linda, in the Japan and Italy and Costa Rica and Greece. In China, and our life is 78 average. Hopefully we can study and figure out globally how we make people live longer. But one of the secret, the mystery or secret for the live longer in this 5 blue zone is minimize stress, moving throughout the day, eating plant-based food. So the secret of longevity is to try to counteract this well-known 12th hallmark of the aging. However, based on our work, recent work, we propose red blood cell as the most abundant cell in the blood and is the most important hypoxia sensor and the metabolic regulator, likely and regulate aging and anti-aging and make people live longer. So in conclusion, my laboratory has studied for almost 10 years from the life beginning, pregnancy, preeclampsia, fetal growth restriction, high altitude, all the way to the aging and the longevity has demonstrated red blood cells very sophisticated hypoxia metabolic sensor can regulate sophisticated its metabolism, trigger the oxygen release, and then regulate tissue metabolic to counteract hypoxia and maintain the microenvironment metabolism and maintain microenvironmental immune and homeostasis. Therefore, we think the red blood cell as a hypoxia metabolic sensor is essential to maintain tissue oxygenation and metabolic homeostasis in disease, aging, and longevity. So future, where we go? We would like to continue study red blood cell and a lot of unknown questions, and then in response to different diseases such as preeclampsia, aging, longevity, and obesity, and etc., even cancer. And I would like to end it with a few slides to introduce the center I have working on. Is in the Xianyao Hospital. It's a National Medical Metabolic International Collaborative Research Center. We have built up the metabolic equipment and also large-scale data analysis. And our center has the talent and young people, creative research team. And we also develop innovative metabolic and lipidomic technology. Particularly, we can accurately use the isotopic metabolic tracer to analyze specific metabolic change. And we also benefit by Xianyao Hospital, also national cohort such conduct a clinical translational study using metabolic screening data analysis to predict the biological aging and also clinical translation. And finally, I want to thank all the people involved in the past and the current. And also, I would like use this opportunity to hope to use our metabolomy serve with every colleague, strengthen our collaboration, and build the future of metabolomic precision medicine. Thank you.
Thank you, Dr. Yang Xia, for your fantastic talk. So, Julie, you are carry over.
Yeah, very fascinating talk and very impressive. Yeah, so now we can go further to the panel list discussion. First, Dr. Sunan Fu, please, yeah, just involve in the discussion. You can unmute.
夏老师是我们的模范了，就是从这个代谢瘤，然后把各种疾病最终都能够联系到一起，真的是特别的钦佩。
谢谢过奖。
真的就说，我是觉得就是能把这一个，就说我们自己也做一点代谢瘤和计算方面的事情，但是要把这些跟就说疾病方面以及跟这个 hypoxia 能够 integrate 在一起，我觉得这是一个真的是一个 masterpiece， 是需要很长的时间去做这件事情。那么 hypoxia 方面我不懂，就是我在听的时候就反过来问，就说